Good morning, everyone. Thank you for coming on this rainy morning. I'd like to welcome you to the 5th edition of the GW ICF Research Evaluation Forum here at GW. And also a special welcome to all the 100 or so individuals joining us through the webinar. And we do have a Twitter account today. It's hashtag GW ICF 2016. And also, there's a chat on the webinar, so if any of you at the end, when we have the Q&A, want to send a question, or you can submit it through the chat line. Thank you. My name is Tom Russo. I'm the Assistant Vice President for Industry Corporate Research here at GW. I'd also like to say a special thank you to our corporate partner, ICF International, who has been so helpful in setting up this event. I want to say it's just a good example for us of how industry and corporates work together. We've been working together with ICF for over 3 years, and one of the collaborations is this very beneficial GW ICF International Speaker Forum. Thank you. Also in the audience today, webinar, a number of government officials I want to recognize from NIH and EPA. All of you, thank you for attending the webinar. It's drier on your end, I'm sure. I would also welcome a new member to the GW family, Dr. Kevin Palfrey, who is the inaugural director of the newly established Autism and Neurodevelopmental Disorders Institute here at GW. Kevin joins us from Yale, where he was the Harris Professor of the Yale Childhood Study Center. Kevin, I just want to please stand up and give a special welcome to him here at GW. And Kevin's labs will be out at our VSTC campus, so, but he will be down here at Foggy Bottom from time to time, we hope. Thank you. So we have a packed room today, and I want to give plenty of time for us at the end to have a good quality Q&A period. So at this stage, what I want to do is introduce our keynote speaker. So what we'll do is we're going to, after our keynote speaker, Dr. Lynn Goldman will, Dean Lynn Goldman will have a panel discussion, and followed by some Q&A. So Dr. Linda Birnbaum is the director of the National Institute of Environmental Health Sciences at NIH and director of the National Toxicology Program. She's a board-certified toxicologist. She has served as a federal scientist for over 36 years now. Congratulations. And has received many awards and recognitions, including Women in Toxicology Elsevier Mentoring Award, the Society of Toxicology Public Communications Award, EPA's Health Science Achievement and Diversity Leadership Award, the National Center for Women's 2012 Health Policy Hero Award, Breast Cancer Fund Heroes Award, and 14 Science and Technology Achievement Awards, which reflect the recommendations of EPA's External Science and Advisory Board. For specific publications, Dr. Birnbaum was also elected to the Institute of Medicine of the National Academies and received an honorary degree from the Ben Gurion University in Israel. Dr. Birnbaum is the former president of the Society of Toxicologists, which is the largest professional organization of toxicologists in the world. She was the former chair of the Division of Toxicology at the American Society of Pharmacology and Therapeutics, and former vice president of the American Aging Association. She has authored more than 700 peer-reviewed publications, book chapters, and reports. She is also an adjunct professor at several universities, including the University of North Carolina at Chapel Hill and at Duke University. She's a New Jersey native. Dr. Birnbaum received an MS and PhD in microbiology from the University of Illinois at Urbana-Champaign. Thank you. Please join us and welcome Dr. Birnbaum.
Well, thank you. I'm very glad to be here. I'm running down from NIH in Bethesda. I kept saying, okay, April showers bring May flowers. And we'll go on from there. But what I want to do today is very, very rapidly and briefly tell you some of the things that NIEHS is doing related to children's health and some of the new methodologies that we're using. So just for those of you who don't know who we are the one institute that is not headquartered here in Bethesda, but we're headquartered in what I used to call the southern part of heaven, and now I remind you that most of our restrooms are now unisex. I if you haven't read what's going on in North Carolina, that's not the good stuff to talk about. But our institute, like any NIH institute, its mission is basic biomedical research. But we have a couple things that make us different. I mean, we do have our Division of Intramural Research, which is doing the basic research. We have a clinical research program. We actually have a day clinic on site where we're doing lots of sample collection. We actually have an asthma clinic, which is looking at the relationship between asthma and microbiome changes. And we have an adolescent clinic, which is looking at the associations between weight gain and breast development in children. We have, most of our money goes out, as any NIH institute does, and we fund lots of programs here at GW. But we also have the National Toxicology Program, which is a cross-HHS effort involving CDC and FDA, but headquartered at NIHS, and it is a problem-solving organization. Similarly, we have funding through the Interior and Environment Committee to fund the Superfund program, which is two parts. Again, problem-solving research, very directed to dealing with exposures at different hazardous waste sites around the country, and worker training and emergency response training. Again, very non-NIH kinds of things, but that we are committed to doing. So we are among the first people involved, whether it's Katrina, whether it was the BP oil spill, whether it was Superstorm Sandy, or right now in Flint, Michigan, working on that. We really have a public health focus. So we redid our strategic plan a number of years ago. I can't believe it was actually released 4 years ago now. But it is really guiding who we are and what we do. And our mission is really to discover how the environment impacts people so we can have healthier lives. And our vision is that we will be providing global leadership for innovative research that improves public health by preventing disease and disability. And the kind of this circle diagram, are these not going forward? Oh, they are there, okay. I'm sorry, I was looking there for a moment, but they are right there. But that circle diagram I have are the themes that we're dealing with, ranging from fundamental research to community engagement to global health. And we used to show that everything overlaps, or many things overlap. There's nothing that's actually in only one area, and that used to turn around, but it actually was there were people with epilepsy who were concerned that it was going to set them into a seizure, so we made it static instead. But so why does environmental health really matter? And WHO last month released a second report looking at the role of the environment in health. And at least 23% of global deaths are attributed to different kinds of environmental stressors. And I think that what you can see if you look at, let's see, I think there is a pointer. Well, anyway, if you look down at the map of the world, you can see that the majority of that 12.8 million deaths, almost 13 million deaths of the year, are in Asia, both Southeast Asia and which would include, for example, India and China, and then followed by Africa and so on. But that's a lot of deaths that are potentially preventable. And many of the causes are chronic non-communicable diseases. It's not infectious diseases other than respiratory. And neonatal conditions play a big role in this mortality as well. And so if we look at some of the different kinds of stressors that you have, I'm just listing, and we'll come back to some of these as examples, whether it's air pollution or water pollution, synthetic chemicals like pesticides or agricultural practices. I'm really not going to talk about climate change, but if anybody doesn't believe that the climate is altering and that is having an impact on health, I would refer you to what the President's Task Force released just 2 weeks ago about the many impacts we're already beginning to see on human health from climate change. And Maria Neira is head of environmental health in the public health division of WHO and said there's an urgent need for investment in strategies to reduce environmental risks in our homes, cities, and workplaces. So what diseases have an environmental component? Well, lots of them. And this is just a partial list ranging from cancer, which is what everyone cares about, birth defects, the other thing that everyone cares about, but then reproductive dysfunction, lung dysfunction, neurodevelopmental and neurodegenerative diseases. We're just really beginning to look at some of the environmental impacts on neurodegenerate, but I'm going to show you a lot of examples of neurodevelopmental disorders, cardiovascular disease, for example, and endocrine disruptors. So the other point is that our exposures are broad. We're exposed to many things. Almost all of our testing strategies look at one compound at a time, and that's not the life we live. We all live in a soup of exposures, whether it's how your diet may modify the exposure to the drug that you took, or the herbal supplement that you had in your food, or, for example, the pesticide that you put to get rid of the of the bugs that were around your house. And if you're concerned about Zika, we're all going to be using pesticides, and we better start looking at the interactions between Zika, the virus, and, for example, the different kinds of pesticides that are being used to potentially affect it. So the point is exposures don't occur singly, and they can either occur at the same time and modify your response, or they can occur sequentially, where the one exposure may modify the other. And we've really begun to look at combinations, and I'm going to come back to the use of the exposome, which is the totality of human exposure that a person would face in their lifetime. So NIEHS research is really focused on understanding the interactions of our genes and our environment. I want to stress with you, there is nothing that is just your genes, and there is nothing that is just your environment. There's a spectrum, and everything is some kind of interaction. Even something like PKU, which is a Mendelian inherited disease, if you avoid the infant being exposed to phenylalanine, the child will grow up at least relatively normally. So in that case, the environmental trigger would be the child having phenylalanine. So it's important to remember, it's not just genes, it's not just the environment. You better be looking at both. So now there are many differential susceptibilities that we have. There's genetic differential susceptibility, there's age differential susceptibility, there's prior exposures. But what I want to focus today is on issues of the pregnant woman, in utero exposures, and exposures of the child during sensitive windows. So we have exposures that occur across our lifespan, but clearly, there are times, for example, the prenatal window is extremely susceptible to change. Childhood is at any time your body is rapidly, or even not so rapidly, changing. Cells are developing, proliferating, differentiating. There are opportunities to throw a monkey wrench. The issue is if you have perturbations that occur during development, they are in general non-reversible, and you can't go back and change them. In adulthood, at least when you're a healthy adult, there's a certain amount of homeostatic balance that you have. That's really not true during development, and it's something that we need to remember. And then when you get to old age is another window of susceptibility. But another window that we need to think about, and I think we're just beginning to focus on, is what about preconceptionally? Can exposures during germ cell development affect the offspring? And if you look at the window of preconception, it's a period of really, really rapid change. You have cells growing, you have cells undergoing meiotic divisions, you have all the different hormonal milieu that has to be present in the appropriate ratios and concentrations. And also what's happening is your epigenome is changing. And I'm really not going to talk much about epigenetics today or epigenomics, but it's important to remember that while your genome may be the score, the epigenome is how it's conducted and how it sounds. So your epigenome controls what genes are turned on when. And that's really all the stuff that we used to say was garbage in the genome. And the fact that 98% of our structural genes are the same between us and the mountain gorilla is because it's the genes that aren't turned into proteins but are controlling expression that are so very important. And that's what the epigenome is. So exposure to germ cells, we know that if you have mutations, you can have teratogenic effects, you can alter the germ quality and fertility, you can have overt DNA damage, and the mitochondria, more and more studies looking at the role of the mitochondria in development as well as in diseases. But in addition, there are a whole bunch of more subtle effects that we're beginning to understand can happen preconceptionally. Things like DNA copy number variations, which can have big impacts on health and disease. Changes in sex-specific gene expression, there can be interferences with meiosis, there can be oxidative stress that can be associated with inflammation, and again, the genome of the germ cells can be changed. And it's likely that some of these subtle effects carry on the offspring, and I can tell you that last week we had a 2-day meeting bringing together our grantees who are looking at this issue of transgenerational epigenetic inheritance. It's not Lamarckian. Okay? But you're changing the way genes are turned on and turned off in a permanent fashion. So I'm just going to give you some examples of preconceptional effect, or exposures that are associated with impacts on the offspring. So I've been saying for a number of years that fathers matter because so much of our focus has been on what's happening to the woman during pregnancy. And some of these studies are in experimental animal models like rats and mice, and I'll tell you which ones are in the humans. But for example, we know there's been a lot of studies now showing that preconceptional exposure of the males in rodents for sure to cocaine and certain other, for example, tobacco, results in altered behavior. We can show that experienced stress is carried on to the next generation following the father's exposure, and that's also in rodent studies. And offspring of the males fed a low-protein diet appear to have impaired liver function. And then if you start switching and you look at human studies and the epidemiology studies, there's more and more data beginning to show that if dads have a high-fat diet, usually obese, that there's an increased risk of diabetes in the father, and has poor semen quality in the offspring, and the paternal obesity is associated with increased risks of autism. And I'm going to have some discussions with Ken about the new autism center you have, because that is a major focus of some of our environmental work. So what about moms? Preconceptional exposures. Again, so much of our focus has been in what happens when mom is already pregnant, but there's growing evidence that maternal obesity alters the maturation of oocytes, changes the redox state of the embryo, and impacts the offspring growth and metabolism, at least in a multi-generational way. Lots of evidence, again, that stress in the mother, in the father, preconceptionally impacts the offspring and leads to altered behavior. And then there's growing evidence, especially, and this is human data, that preconceptional smoking is associated with an increased risk of congenital heart defects. We know that smoking during pregnancy is associated not only with small for gestational age babies, but impacts on IQ and small birth weight, but then the children become obese by the ages of 8 to 10, and that persists in a higher risk of diabetes. And then there's also from the very exciting cohorts that Jermaine Buck-Lewis from the National Institute of Child Health and Disease has studied, where she's been looking at paternal and maternal exposures to POPs. Those are persistent organic chemicals, things like DDT, PCBs, are associated with low birth weight in the children. Again, preconceptional exposures. So what are the exposures during pregnancy? Well, it's kind of the laundry list that I referred to before that in the WHO report. We're talking about everything from contaminated food and water and household products like pesticides and cleaners to chemicals and electronics, prescription drugs, cosmetics. I've mentioned stress, and I'll mention it again, agricultural chemicals, and then anything that's synthesized. But anyone who thinks that what you eat in your food is not chemicals and that's somehow inherently different than synthetic chemicals is a little confused, because in fact they're just all chemicals, and what you eat and the composition of what you eat really will impact your response to various chemicals. Some of that's going to go via the microbiome. Some of it may just be a change in your inherent metabolism. So I've already said that early life exposure it occurs at a time when the body is rapidly changing, and the early life exposures can have long-lasting effects. And this is the whole basis for a field which has really been in existence now for only about 10 years, which is the developmental origins of health and disease, or DOHAD. And this is where early life exposures, and I almost prefer to use that term because that includes preconceptional and conceptional and childhood and so on, can lead to epigenetic programming changes that alter when genes are on and off. Your metabolism, when certainly in utero, as well as in the infantile period, is not the same. Your ability to handle exposures, your immune system is not functioning the same way, makes you more susceptible to exposures. And the changes that occur during development, as I said before, permanently alter the potential of an organ. And so you can either directly increase the sensitivity or alter the set point for disease that may occur later on in one generation to the next. So this developmental origins of health and disease, there can be a variety of kinds of exposures that lead to alterations in hormonal regulations and metabolic pathways and cellular differentiation. That can be expressed by changes in your epigenome, I didn't say before, but the epigenome, what it is you have the same DNA-based sequence, but you may put little methyl groups onto the DNA, which tells it whether to be on and off, and then the DNA doesn't exist as that long strand of spaghetti in your cells, but is actually curled up and folded into a three-dimensional structure called chromatin, and that involves histones, and the histones can be methylated, acetylated, propionated, ubiquitinated, butyrylated. Can have different groups put on them and different combinations, and again, that affects how they how the chromatin fold up, which impacts whether genes can be turned on and off. And then in addition, some of those RNAs running around the cell that we used to say were junk, the little microRNAs and the other non-coding RNAs, are absolutely critical for, again, controlling expressions of genes. And we're now finding, this is an epigenetic mode of inheritance, that these non-coding RNAs are present in sperm and are actually transferred to the egg at fertilization. So it's another way for the dad to have a role from preconceptional exposure. And anyway, these alterations can lead to long-term effects and increased disease risk across. So now I'm just going to go through a bunch of kind of examples from some of our grantees and others related to some of the kinds of exposures we have a lot of concern with. So prenatal PAH exposures. PAH, yes, when you barbecue your steak, you know, the drippings generate PAHs, but among our biggest sources of PAH are combustion of organic material, and diesel exhaust, and especially diesel trucks, are a big source of PAH exposure. And what you can see here is whether you're looking at children at age 5 or age 7, their BMI increases relative to their maternal exposure, to the maternal exposure in utero. This is based on cord blood levels. So that's both maternal and child's exposure, but in utero exposure to PAHs. And it's a persistent change. We know that early life exposure to phthalates, another class of major products that are in personal care products, they're in your food packaging, they're in all kinds of makeup and nail polish and so on. And we've known for a while that there was actually an association between early childhood exposure, exposure to the child, and their susceptibility to asthma. But what you can see here, that at least for a couple of phthalates that were looked at here, these are the metabolites, the monobutyl phthalate and the monobenzyl phthalate, which are the products of the diambutyl phthalate and the butylbenzyl phthalate, that these phthalates are associated with an increased risk of asthma following in utero exposure. So again, mom's exposure associated with what you see in the offspring. Now, this is actually not looking at what you see in the child, but there's a very interesting study that's been going on for the last 30 or 40 years through our Superfund Center at Berkeley, and this is actually studying an unfortunate poisoning that occurred in northern Chile, in Antofagasta, Chile, where the river that was the water source for the town was used to wash out the mine tailings in the copper mine upriver from the town. And this led to a major arsenic contamination that occurred for about a 20-year time period before they realized they were having people with frank arsenic poisoning being detected in the town. But what the really interesting thing here is what you're seeing here, both for lung cancer and bladder cancer, is there's a dose-related increase in related to how much arsenic the exposure, but this is also showing you an age-related change, so that the people who were exposed, because we know when the pollution started and when the pollution stopped, we have people who were only exposed in utero. We have people who were only exposed, say, in childhood. We have people who were only exposed to adults. And what's been shown here is that the people who were exposed in utero or in early childhood 50 years later have the highest incidence associated with their early life exposure. Much higher incidence from early life exposure to arsenic cancer than if exposure occurred as an adult. I think that's something important to think about, and I can tell you for experimentalists, it's something that we're spending a lot of time thinking about. So just to briefly, arsenic we all know is a bad thing. Not all the effects I have listed here have to do with childhood exposure. The bronchiectasis, which is a lung disorder, has been shown. In utero, early life exposure is associated with bronchiectasis by age 20. For example, there are lots of reproductive and developmental problems, neurological problems, and a study coming out of school children in Maine has shown that those children who have been drinking their whole lives water in wells within the EPA and WHO regulatory limit, but above 5. The regulatory limit is 10 ppb, but above 5, so between 5 and 10, have lower IQ scores than children below 5. And I think that's an important thing to understand, that effects are happening at very, very low doses. I'm beginning to believe that arsenic, like lead, there's no such thing as a safe level. We also know that there are associations with type 2 diabetes and lots of mechanistic studies about how arsenic likes every endocrine receptor that you ever knew about. Now, I also wanted to talk about another neurodevelopmental impact, which is ADHD. And with pesticide exposures, both in animal models and in humans, we're finding that certain pesticides, especially the organophosphates like chlorpyrifos, just use chlorpyrifos as an example, actually banned for residential use in 2002, but continues to be used in agricultural uses. So if you happen to live near a farm, you're going to be exposed. But we're finding that those children born to women who had elevated chlorpyrifos exposure have lower IQ, more behavioral disorders, and new data showing an increased risk of autism. And I want to say that some of the power of these studies are going to come out of, I'm going to show you our children's centers in a few minutes, and the point there is we had 3 different cohorts, a Mexican-American cohort, a Dominican-American cohort, and a Caucasian-American cohort in 3 different locations, and we found essentially the same associations between IQ deficits and exposure to chlorpyrifos in utero in all 3. And I would call that a replication of the finding. So, there are lots of heavy metals that are out there that we need to be thinking about right now. So much of our concern, if you look at the bottom bullet, is showing that very low levels of lead are associated with lower IQ scores in children. Again, no safe level of lead. We're all concerned about Flint, which is a clear example of environmental injustice, but Flint is just the tip of the iceberg about problems of lead contamination in many inner city areas and Rust Belt communities, as well as poor rural areas around the country. But other compounds, I've mentioned the arsenic already. We know that methylmercury showed decreases in IQ from exposure. I mean, there's the long-going study in the Faroe Islands following the children there with methylmercury, different levels, and looking at it from seafood consumption. And at age 22, the IQ deficit still persists. So again, as I said, what happens during development doesn't reverse. And then more and more effort being put on looking at exposure to manganese, which we know high levels of manganese in adults is associated with effects on your nervous system. But now we're finding that exposure in utero or to children is associated with poorer memory and attention, even at very low levels. Now, what about air pollution? We're all exposed to air pollution all the time. We look at the pictures of Beijing and Delhi and Agra, and we think this is so terrible. Our levels aren't that high, thank goodness, but our levels are still being associated with adverse effects. And we know that living within 75 meters of a major roadway is associated with increased risk of asthma. It's also associated with decrease in IQ. There are now 12 studies showing that near roadway air pollution is associated with, this is again to the mother, or in the first year of life, is associated with an increased risk of being on the autism spectrum. Air pollution, again, like lead, like arsenic, I would say, probably no safe level. And this is whether you're talking ozone or PM or nitrous oxides, all problematic, and also impacts on the immune system as well. So we focus on the outdoor air. But you know what? Our indoor air is much worse than our outdoor air. And in many parts, especially the developing world, where people cook with biofuels in small huts, the levels inside the huts are so thick, I would say, with PM and black carbon, that they're worse than what occurs even in Delhi or Agra, for example, on a very bad day. We know that there are over 3 billion people exposed each year to especially indoor cook smoke. Most of them are women and children. We know that more than half of premature deaths among children under 5 are due to pneumonia, which is associated with the particulate matter inhaled from household indoor air. We know that there's decreased neurodevelopmental performance associated with wood smoke exposure. We know that the biomass fuel burning is associated with an increased risk of low body weight in the children and respiratory illness and infant mortality. And we know that asthma is associated with open fire cooking in Venezuela. Unless you think this is only an issue in the developing world, let me tell you that 16 million people in America cook with biofuels, usually wood, inside, and not necessarily with proper ventilation. So back to phthalates a minute, and I want to switch to a little later exposure, not in utero or infantile, but what's happening around the time of puberty. And this is a recent study that, again, has come out of our breast cancer and the environment research program, where we've been looking at exposures that are occurring to young girls as they go through puberty. And what we're finding here is that exposure to certain of the higher molecular weight phthalates is linked to a later pubertal onset, and this is especially true if you look at the darker lines in girls of normal weight as compared. There's still the impact in the girls who are overweight, but it's less noticeable than the girls who are of normal weight. So in this case, the endocrine disruptor phthalates are leading to a decreased rate of puberty or slower pubertal onset. So what is this all costing us? Well, Leo Trisandi of NYU, who has been doing a number of different studies, actually estimating the costs yearly of these, and he's done this in conjunction with the Endocrine Society and the European Union, and he has recently estimated that in 2008, the cost of environmentally attributable costs of childhood lead exposure, methyl mercury exposure, developmental disabilities, asthma, and cancer was in the neighborhood of $71 billion. Now, he has also estimated in another study that if you looked at the total impact, the cost of lost IQ from early life lead exposure worldwide is over $1 trillion a year. I think these are big numbers, and right now the only thing that really talks on the Hill is money. So these are figures that are helpful to have. So what is NIEHS doing? Well, we're spending approximately a quarter of our budget dealing with issues of children's health. The sizes of the bubbles here really reflect how much our effort is in these different areas. So you can see that neurocognitive outcomes is a major focus of our child health research program. Cancer, birth defects, respiratory outcomes, metabolic outcomes. Pretty much we have to cover the waterfront, but it's about a quarter, so we're spending upwards of $150 million a year focusing on issues of children's environmental health. And this is just an example of where we have funded, many of them co-funded with EPA, Children's Environmental Health Centers in many places around the country, where we started this program in 1998 in response to the President, the Clinton call, executive order related to children's environmental health, and we continue to fund programs around the country. What is so important in these studies, and these involve both basic mechanistic studies and epidemiological studies. And what's really important is when you do the epi and you can develop longitudinal cohorts that you follow from, we don't have any preconceptional cohorts, I'm sorry to say, but we do have lots of pregnancy cohorts and birth cohorts. Our oldest kids, who started back around 1900, are now getting to the age where we expect some of them to start having children soon. So we're going to be able to look at second-generation effects in some of these cohorts. And I want to very briefly just tell you about one of the new approaches that we launched at the end of last year called CHEER, or the Children's Health Exposure Analysis Resource. And this is a $50 million effort for the next 4 years, which will be supplemented by funding that we'll be getting from the new ECHO program, which is the Environmental Consequences of Health, Environmental Causes of Health Outcomes, I think that's what it is, which is not the National Children's Study, but is the reformulated program that people will start. There are 160 applicants to this program, and we expect the funding to be done by September of this year. And we just announced that Matt Gilman of Harvard will be the ECHO program director, and we're very happy about that. But the CHEER program is the first analytical exposure network that's been set up around the country. So we have six laboratories to which anyone that has pregnancy or children samples, and I would also say preconceptional samples, that they would like to submit for analysis, they can, if they have NIH funding, they don't have to pay, they just submit them to the 6 laboratories. There's a data coordinating center which will do all the, you can't just look at your numbers anymore. You got to crunch it, and you need bioinformaticians and, all kinds of the complexity of the data that we have today to analyze it. And there's a coordinating center. So there are basically 8 groups that have been funded. the coordinating center, the data repository, and the 6 laboratories. And the laboratories will be doing what I'll call, and I'd say in quotes, standard biomonitoring, because like CDC does for known analytes. They'll be looking for response into early markers of response, measures of oxidative stress, inflammation, things like cytokine release and so on. And then they're going to be developing methodologies and moving forward on exposomic approaches. These are agnostic approaches where you just look at everything that's there. You might have 20,000, 30,000 different peaks in your high-resolution chromatograms that you're getting from different kinds of mass spec or even MNMR, and you're going to be looking for patterns and trying to identify things that, stop looking under the lamppost and look at what else may be out there. So we're very excited about this resource. So where are we? Related to children's healthy environment, we have robust data that prenatal time period is critical and likely the most important window of susceptibility. And childhood is also a window, at least for some diseases and disorders. There's emerging data that preconception is a sensitive window in males and females. There's preliminary data that, and people haven't really looked at this very much. How susceptible is the mother herself, not her fetus, but the mother, to exposures during pregnancy? Her body is rapidly changing. Certainly we know that, for example, if a woman develops breast cancer during pregnancy or shortly thereafter, it's usually a very aggressive form of breast cancer. Is that related to what was going on during pregnancy? We know that there are multiple windows across the lifespan. We haven't looked at all those windows of susceptibility. And we need to remember that a given exposure is going to cause different things in different critical windows. And because your susceptibility varies. So I want to thank you all and remind you that a good start lasts a lifetime, and you can't change your genes, but you can change your environment. Thank you.
Linda, thank you very much. It's always a pleasure for me to welcome today's moderator to this event, Dean Lynn Goldman. Lynn Goldman is the Michael and Laurie Milken Dean of Milken Institute School of Public Health at the George Washington University. Before joining Milken Institute in 2010, she was a professor of environmental health sciences at Johns Hopkins University's Bloomberg School of Public Health. Prior to that, Dean Goldman served as assistant administrator to the EPA's Office of Chemical Safety and Pollution Prevention, where she was responsible for national implementation of chemical and pesticide safety and pollution prevention laws. She also worked at the California Department of Public Health, where she headed the Division of Environmental and Occupational Disease Control. Dr. Goldman serves on the board of the Environmental Defense Fund and is a member of both the Institute of Medicine, where she also serves as a member of the Governing Council, and the National Academy of Sciences Board of Environmental Sciences and Toxicology. In 2013, Dean Goldman received an honorary doctorate from, is it Örebro? How do you pronounce it? Örebro, thank you, University in Sweden, for her contribution to chemical legislation in the U.S. and Sweden, and her influence on the research conducted by the University of Nanotechnology Environmental Research Center. She holds a B.S. in conservation of natural resources and an MS in health and medical sciences from the University of California, Berkeley, and an MPH from Johns Hopkins University, and an MD from the University of California, San Francisco. Lynn?
Thank you. Before I introduce our panel, I need to give my personal thanks to Dr. Birnbaum for such a wonderful kind of romp through the whole field of environmental health in children, and particularly the fabulous work that is going on at the NIEHS. I think that Dr. Birnbaum is an example of the important role that leadership can play in the government. She's been a passionate advocate for not only the protection of the environment and for public health, but also for children specifically. And I think it's clear that she's making an enormous difference. Thank you so much, Linda. That was wonderful. So we're going to have a panel that's going to have a discussion here. And the way we're going to do this is that we're going to hear a little bit from each panelist. We're going to have a little bit of time to have a discussion among the members of the panel, and then we're going to include the audience in that process. But I think it's obvious from what you've just heard from Dr. Birnbaum that we're in a truly exciting era in the field of not only environmental hazards to children, but really biomedical science writ large. The exploding knowledge in areas like epigenetics, our understanding of the interplay between our exposures, environmental exposures, our genetics, our epigenetics, hormonal signaling of all kinds, and the microbiome, which he slightly mentioned. It's not only that we're beginning to understand much more about health and disease, we're just beginning to understand what we really are as human beings. And a major part of that has to do with understanding what's going on with children, because children are not only important in and of themselves, and I would say that they're extremely important in and of themselves, but they're also important because of what they become and because of the fact that so many health hazards are imprinted in them over their lifetimes, as you heard from Dr. Birnbaum. And our three speakers are very prepared to tell us about some of the aspects of that. Dr. Birnbaum already mentioned the importance of the people who work to help us to understand these huge volumes of data that we now can produce. And we have as our first panelist Jonathan Cohen, who in fact is an expert of that. Dr. Cohen is technical director at ICF, where he has 33 years of experience in statistics and data analysis. He has a PhD from Imperial College in London with an emphasis on the areas of applied probability and statistical aspects of extreme value theory. He has been a professor in various institutions, but he has actually been at ICF for 26 years, where he manages a wide variety of projects for the government and commercial clients. It's particularly projects that involve the application of statistical data analysis to environmental, land, air, water, and emissions data. We also have with us Ami Sota, who is an assistant professor in our Department of Environmental and Occupational Health at the Milken Institute School of Public Health. Dr. Zota is an environmental epidemiologist, and her interest is in formulating hypotheses and studies that will help us to unravel how some of these very complex issues related to not only environmental exposures, but also social disadvantage, and in terms of disparities in reproductive and children's health across the life course, as well as pathways that might lead to ways to prevent some of these adverse outcomes. Her research has been featured in the Washington Post, Time Magazine, the Atlantic Monthly, and so forth. She currently has a K99R00 Career Development Award from the National Institute of Environmental Health Sciences, for which we thank Dr. Birnbaum. Thank you very much for that. And under this grant, she is developing conceptual and methodological frameworks for examining the independent and potentially synergistic effects of endocrine disrupting chemicals and psychosocial stress on perinatal outcomes. Our third speaker, Dr. Rebecca Clifton, is Associate Research Professor of Epidemiology and Biostatistics, also at the Milken Institute School of Public Health, but also at the university's Biostatistics Center, which she joined in 1997. Dr. Clifton has been a principal and co-investigator on a number of very large multi-center and multi-cohort collaborative studies. Her research interests include gestational weight gain, preeclampsia, preterm birth, and the impact of intrauterine environment on child health. She has served as a grant reviewer for multiple NIH institutes, as well as obstetric and obstetrical journals. I think it's my job to bring the clicker down to our first speaker. Thank you, lead off, John.
The button, the green one? Nope. This.
I think actually you point that way. Point that way.
How do I turn the? How do I do the next one?
It ought to be going. Let's see. This big one. Yeah.
Oh, the green one. That's right.
Yeah.
Okay.
All right.
That's me. Okay. So I want to talk about the America's Children and the Environment project, which I've been managing for EPA. There's the websites there, epa.gov/ACE, where you can find all the details. And what we're looking at is measures of and indicators of children's environmental health. And we summarize the available data, we look at trends, we look at demographic differences, and we have various indicators that we're looking at in three groups. First of all, we look at exposure. We look at air pollution, drinking water exposure, pesticides in food, contaminated lands. That's the percentage of children living close to a hazardous waste site. So looking at children's exposures to environmental contaminants. Then another set of measures we're looking at is to do with biomonitoring data, essentially dose, but there's a translation between the biomonitoring data and the actual dose, which is non-trivial. But we're looking at concentrations in both women of childbearing age and also children for lead, mercury, cotinine, phthalates, BPA, perchlorate. There's the list up there. So summarizing trends and demographic differences in the concentrations. And then the third set of measures is to do with the health outcomes for children. The percentage of children nationwide that have current or asthma, other respiratory diseases, cancer, neurodevelopmental diseases like learning disabilities or ADHD, obesity, of adverse birth outcomes. So the outcomes of the environmental exposures. The report that is at that website summarizes the trends and some of the demographic differences for different groups of children. But one interesting thing that we do is we're often interested in exposures, in utero exposures, and for the biomonitoring data, what we have is data on women of childbearing age. And one sort of novel idea that we've applied is to adjust for the fact that younger women are more likely to get pregnant. There's more births to younger women than older women. So in order to estimate in utero effects, we reweight the data from the NHANES study based on the proportion of women or proportion of births to women of different age groups or different races and ethnicities. So I think that's a useful idea of estimating in utero exposures, potential in utero exposures, when all you have is data on women of childbearing age.
Thank you. Now, let me get this. Okay, so you press the green button, right? Yeah, the top one. Yeah, okay, you are next.
Okay, so that's me. So my work really occurs at the intersection of environmental exposures, social disadvantage, and health disparities, both understanding pathways of intersection, but also identifying new pathways to prevention. And my work, the research questions I ask roughly fall into 4 categories. One is identifying sources of exposure to these chemicals. So as evidence accumulates about the adverse health effects of some of these chemicals, such as phthalates and flame retardants, increasingly regulatory, authoritative, clinical bodies are recommending exposure reduction. But clearly there are few options available to people like pregnant women on how to actually reduce their exposure. So it's very important to identify modifiable sources. Last week, I actually had a new paper come out showing that fast food intake may increase exposure, population exposure to high molecular weight phthalates. And this was actually done leveraging the same data set that Dr. Cohen spoke about, the NHANES. And NHANES has really been an invaluable data source to the environmental community because it's roughly representative of the U.S. population and really has a wealth of information on exposures, health outcomes, and other medical and health characteristics. Another area of my research is really focuses on advancing exposure assessment during pregnancy and early development. Dr. Birnbaum very convincingly discussed the importance of this vulnerable period of development. So it really, our understanding of exposures during this time will really help our evaluation of health risk. So in both looking at how chemicals partition across the maternal fetal unit during various points in pregnancy, including looking at pregnancy tissues, placenta, and also starting to develop new methods to characterize cumulative exposures. We increasingly know that we must do a better job of accounting for the chemical soup. And in terms of even looking at vulnerable populations, those people who may have, that have the cumulative burden may be different than people who have higher exposures to one or two chemicals. So really, we need better methods for looking at chemical mixtures and cumulative burdens. Another area of my research involves using analytical methods to understand the intersection of chronic psychosocial stress and environmental chemicals, because socially disadvantaged communities often experience a higher burden to both of these. So I look at both biomarkers of early response, such as telomere regulation during pregnancy, as well as overt health outcomes like pregnancy complications. And lastly, because we all ultimately want to improve public health, I think it's important to evaluate the regulatory and policy actions that have taken place, and to evaluate the impact of these measures on exposure and health reductions. And once again, I've leveraged the NHANES data sets, for example, to show how NGO activity as well as federal regulations have changed Americans' exposures to phthalates, albeit it has led to the rise of problematic substitutes, which is another reoccurring problem in our quest for better environmental health. So I'll leave it at that.
So I'm just going to briefly describe two studies that we've been involved in. These are studies that recruited women during pregnancy, and we modified the intrauterine environment and then followed the offspring out to see the impact that those changes had on offspring health. So this first study is a consortium. It's an ongoing consortium of 7 randomized controlled trials of lifestyle intervention versus enhanced standard of care in women with overweight or obesity. So this is an ongoing collaboration. We just finished recruitment in December of this past year, with our last deliveries expected this summer. And then we'll be following the children out to 1 year. The goals of these trials are to reduce excessive gestational weight gain. So both preconception obesity and excessive gestational weight gain are established risk factors for childhood obesity. So we are attempting to stop that cycle. And so in this study, these cohorts are racially and ethnically diverse. About half of the cohort has reached their 1-year follow-up, and so far we have very good follow-up of our children. So the study, as designed, is funded to follow the children through the age of 1, both the mothers and the children through the age of 1. Focusing on the child outcomes, we are looking at body composition, so we're measuring weight, length, and skin folds. Now, recent data has suggested that the prenatal effects may not become evident until the age of 2 or later. And so we understand that we need to have a longer-term follow-up. And so we have submitted funding to follow these children through the age of 7. And this would include the measurements that we completed at birth and at 1 year. So that would be the weight, height, skin folds, and then also adding more sophisticated measures of body composition, so air displacement plasmography and MRI to get a better estimate of body fat. We are also going to be measuring cardiometabolic factors, obviously given the relationship of those with obesity, and looking to see if there is an impact of lifestyle intervention administered prenatally on infant health. So the second study was recently completed, and these studies came about in the late 1990s. There were studies that were published that showed women that had elevated TSH or were classified as hypothyroxanemia during pregnancy, their offspring resulted in a lower IQ. So in the late 1990s, this resulted in many professional organizations endorsing the screening and the treatment of pregnant women for subclinical thyroid disease. And so what we wanted to do was to design studies to really evaluate this impact on child health. And so 2 parallel randomized controlled trials were conducted. The first one among women diagnosed with subclinical hypothyroidism, and the second one among women diagnosed with hypothyroxanemia. They were randomized to either thyroxine or placebo, and the infants were followed for 5 years. These results were just published in February of this year, actually presented in February this year at the Society for Maternal Fetal Medicine. And the studies found that there were no differences in neurodevelopmental testing that was conducted annually, but most importantly, that there was no difference in IQ at age 5. Further, there was no difference in behavioral testing, including ADHD, that was assessed at year 4. So these studies show that that treatment of subclinical hypothyroidism and hypothyroxanemia is not warranted. It did not improve cognition or behavioral measures in the offspring through the age of 5, and thus the screening of pregnant women for subclinical thyroid disease is also unwarranted.
Thank you very much. I think that one of the things that I find interesting about these examples is the applicability of this kind of research to real-world decisions, whether it is standards of care for medicine and obstetrical care in particular, which we don't deliver perfectly well in this country. I think many people in the audience are aware of that, or whether it's environmental regulation and an agency like the EPA being able to actually monitor whether the regulatory system is producing the results that it's trying to produce by looking at the trends or not, or to actually be able to understand which chemicals need to be regulated in the future and whether our regulation is actually wise in terms of targeting the right compounds, the right things to regulate. I guess a question that I want to lead off with, and I think it's going to be a fairly difficult question, but that's me. But and somewhat inspired by a workshop actually that Dr. Birnbaum and I participated in about a year ago. And that's a problem that we're beginning to appreciate, which is that many of the chemicals that Dr. Zota studies and that Dr. Cohen has been monitoring for the EPA actually seem to have a potential to impact on obesity, the problem that Dr. Clifton is studying. And in fact, that we're beginning to see that it's an array of the very same chemicals that Dr. Birnbaum had presented that seem to be triggering hormonal changes that are not favorable in terms of our metabolic state. And how do we bring together the data that we need to bring together to look at a problem like this when we have not, your research on obesity hasn't really monitored the chemicals, your research on chemicals hasn't really looked at this problem of obesity. We've kind of tended to be in different silos, and that's even true in the National Institutes of Health, that obesity kind of sits in the NIDDK silo, and obstetrics sits in the NICHD silo, right, and environment in the NIEHS silo. And perhaps start with Dr. Cohen and move my way to Dr. Birnbaum, kind of from right to left. But
well, I'm guessing that one problem with obesity is that one reason why people are obese is probably they're eating food that isn't good for them, and prepackaged food, I don't know why I'm saying this because I'm probably an example of it all, but nevertheless, prepackaged food has nasty things in it, and so you're going to get worse environmental exposures just from the sort of food that you eat.
That's an interesting point, and it is very true, and I should have said it right out front. It is very true that we know that a major factor is, of course, that we have an unfavorable energy balance across our population, in that we are taking in too often more energy than we're expending. However, this evidence says, yes, that's true, and at the same time, these chemicals really are having these hormonal impacts. Ami?
I think to further that, I think too long environment and nutrition have been siloed, and as Dr. Birnbaum said, there isn't that much difference between the chemicals that we study as nutrients and the chemicals we study as unintentional contaminants. And I think obesity in particular is a disease that really requires integration of these two disciplines, whether it's those expertise coming together in a cohort study, but as well as at the experimental level to understand, how fat versus carbohydrates, what does that do to the metabolism or transport of these chemicals in the body.
Now, you recently had a study that actually showed what Dr. Cohen was hypothesizing in terms of the fast foods and chemicals are associated, correct?
Right. So we found 20 to 40% difference. So people who ate a high amount of fast food in the prior day, there's people in the U.S. that are eating 35 to 100% of their calories from fast food. These people had 20 to 40% higher levels of these phthalates. And interestingly, we also looked at whether it was driven by the fat in the fast food, and that was part of the picture, but not the entire picture. That's interesting because the phthalates are coming from this food that may be a risk factor for obesity, but then phthalates themselves may also be a risk factor for obesity.
Rebecca, did you?
Yeah, I would just add that I think it's really important to continue cross collaborations across institutes. So the consortium that I described, looking at lifestyle interventions, that is NIDDK is the primary institute, but we also have funding from NHLBI and NICHD. And so I think it's important to bring these groups together to evaluate that, and then also to have rigorous means to assess diet, and to also collect samples. So I know for all of our projects that we're collecting blood and urine and cord blood and placenta so that we can look at exposures to other chemicals.
When you saw Dr. Birnbaum present the cheer thing, is that a kind of an opportunity that you could use? It looks to me like it could be a potential avenue to get some of that exposure measurement done.
Absolutely.
Absolutely. I'm never going to give anyone a buy on eating too much and not exercising as part of the contributors to obesity, but I don't think it's the whole thing. There's a whole new science of obesogens, which are chemicals which basically alter your body set point. And early life exposure, there are a large number now of experimental studies using animal models, and there are human studies that are showing associations with certain kinds of chemical exposures and elevated body weight. For example, in children, which once you are an overweight child, you often become an overweight adult. So smoking is one of the best examples, and I mentioned that before, that in utero exposure to smoke is associated with obesity. There have been, a couple years ago, it was 23 studies, which were also the association with overweight, obesity, and type 2 diabetes. There have been more studies since then where they followed the children who, at one point, they had children who were 8 to 10, and now they have people they're following at 20 to 30. We know from some of the studies of nutrition, if you go back to the hunger famine that occurred in Holland in 1944, when there was nutritional deprivation in the Dutch people. The Dutch famine is actually what they call it. And those not only the people who were in utero at that time, but when they grew up, they had a higher risk of obesity and type 2 diabetes, cardiovascular disease, and cancer, and now we're seeing overweight and obesity in their children. It becomes very difficult to disentangle some of the what's going on. We think this is an epigenetic association that we're seeing, but we have lots now of environmental studies of different kinds of environmental exposures in utero, which are being associated, and some I think I even mentioned preconceptionally, which are associated with overweight and obesity in the offspring. And I really, I don't think you can blame a young child for their overweight. I do. Can I raise another question? I want to challenge Rebecca a minute about her study with the hypothyroxanemia and the hypothyroid. And you treat that once the women show that they might have their elevated TSH. That may be too late. And so are we beginning to recommend, I think a lot of our recommendations of things to avoid, for example, or treatments to give are occurring too late. So we know, for example, that the addition of folic acid to food supply has resulted in a very significant decrease in open neural tube defects. But we're beginning to find that folate may actually be preventative, for example, or reduce the risk of autism. But when is that happening? Well, it may be happening preconceptionally and up through the first 8 weeks of pregnancy. And that's not the time that a woman has gone to her doctor and started taking pregnancy vitamins. So we need to begin to focus, I think, more on men and women of childbearing age and improve their health status in order to have healthy children in the future.
So excellent point. And one of the things that certainly did happen with the folate is that when the science really came in to say you need to have folate sufficiency even before a woman becomes pregnant, that at the time of conception that she needs to be in folate balance. That was why the decision was made to supplement the food supply rather than to use a pill. Because once you realize you're pregnant, it can be too late. A lot of people don't recognize pregnancy the very day it happens. Other people do. I realize that. Some of you in the room probably, But I think that for most people, by the time the pregnancy has been diagnosed, if you don't have enough folic acid, it's too late. And it is certainly true, and it's something we don't talk about very much, that many people in our country don't have enough iodine in their diets. Because one of the things that is true, that the public health approach to iodine was to supplement table salt, but if somebody has 100% of their calories coming from fast food, like some of the people in Dr. Zota's study, they're not even using that. And there isn't necessarily iodine in many of those processed foods. And there is data that there are people, even in this country, who don't have enough iodine, don't have enough vitamin D. It's crazy. But I don't know if you have a comment on that. I mean, what kind of study would you have to do in order to, you would need a preconception study.
Right, and I think I definitely think that we need to shift towards preconception. It's a very big challenge because I think 50% of pregnancies are unplanned. And so I think that creates a very big challenge. For the studies that we have done on pregnant women, it's extraordinarily difficult to recruit them early in pregnancy. We try to focus on that first prenatal visit, but there are a lot of women, especially those low SCS, that do not come in for prenatal care until 20 weeks. And so it's one of our challenges that we have as far as trying to get the interventions, whether it's the lifestyle interventions or treatment of some form early in pregnancy to actually make a difference. So no, it's definitely a challenge.
So I was wondering, Linda, what you were going to say when you brought that up, because it's something that occurred to me, and that is that does the synthetic thyroid hormone actually correct the thyroxanemia? Does it actually correct it? Because especially if you're seeing a higher TSH, what is that actually indicating? And because if you're seeing that and it's because you don't have enough iodine in your diet, you're not making thyroid hormone, I think you can correct it by replacing it. But I think if you're seeing it because you're exposed to an environmental contaminant that's blocking a receptor and causing the production of TSH, that's still happening,
Right.
whether you give the synthetic thyroid hormone or not. And so I think that's another consideration, at least for elevated TSH. Why is that happening? It could be you don't have enough thyroid hormone around, but it could be because the receptors are blocked and there's just there's a disruption of the thyroid system.
No, I totally agree. But I really think the point I was making even more is going back to the issue of critical windows.
Right.
And we know that so much of the role of thyroid in neurodevelopment is occurring very, very, very early. And that's true for other things as well. I actually took out a slide, because I had too many as it was, where I kind of looked at the different developmental windows, and things that are happening within the first 8 weeks of pregnancy. The heart is formed, totally formed then. And so if you have something which is a cardiotoxic drug, it's probably not going to have much of an, or would affect cardiac development. You have to have exposure earlier in order to impact it, because you've passed that window. So we have to think about when are the susceptible periods. And again, the point that I tried to make is that different things happen in different windows. So, a given, say, chemical exposure like the phthalates may do one thing preconceptionally, they may do something else during the first trimester, something else later on, something else in infancy. And we somehow have to begin to look at all those factors.
Another point I just wanted to add, I know you focused on neurodevelopment, but the thyroid gland is particularly vulnerable during pregnancy. Pregnancy places a high demand on the thyroid gland, and that can also have repercussions for the woman independent of the child, right? So thyroid fluctuations, people are suggesting it's a risk for postpartum depression, which clearly affects the child, in another way, as well as chronic disease in the woman later in life. So I think there are also other endpoints that may be relevant to women's health from thyroid disease during pregnancy.
So I want to shift gears a little bit. We heard a little bit about how environmental data that EPA collects are available on the web, and we've heard a lot about exposures that we can measure directly in people, whether in blood or urine or otherwise. But there are a lot of exposures that are very difficult to measure, such as exposure to air pollution. And although some people have succeeded in putting devices on study subjects that they carry around in backpacks and so forth, normally we don't have that kind of information, and certainly for something like NHANES, we don't have that. I think for very large studies, we'll never have that unless it could be made a lot smaller and cheaper, a lot smaller. But how can we use really the wealth of data that are available to help to impute exposures for people, for things that we can't take direct measurements? What are your thoughts about that in terms of the statistical challenge of doing that? Take air pollution, for example. There's so many measurements, and what do we do? How do we decide what people are actually exposed to?
Well, as you say, that there have been quite a few studies where they actually do have people, with an air exposure, monitor on them, and they walk around and they, you measure their exposures in different microenvironments, indoors and outdoors, and then they can be used. So that data is useful, and there are several models that take that data and sort of model somebody's exposure over a longer period by following them from microenvironment to microenvironment, taking into account their activity patterns. So,
So in fact, air pollution, I think, is something that we do a better job of. Outdoor air pollution, a better job of managing. Since indoor air pollution isn't regulated, nobody measures it unless they're specifically looking. But outdoor air pollution, we have different kinds of, we have stationary monitors that are set up that EPA has in a vast database, and that's the kind of modeling then you can impute what people might be exposed to. You also have mobile monitors, and then you have the personal monitors. And we're getting better. We can now have do air pollution monitoring, and the monitor is the size of a deck of cards. So it's not something that is terribly difficult or terribly invasive to wear. We also have developed, Kim Anderson at Oregon State has done a great job of developing these silicone bracelets, basically, wristbands that come in different colors. We actually have been trying them out in the field in some of our children's cohorts. The only problem is the kids don't want to give them up at the end of the study because they like these color bands. So we now substitute bands that don't have the sensors in them. But they can pick up 100 different kinds of volatile organic chemicals on these wristbands. So I think this is a very powerful tool, especially if you want to do some kind of longitudinal exposure and follow what's going on. So I don't think it's not doable. I think in very large population exposures, you're going to have to use the different models that have been developed to predict, at least in a given area, what the exposures may be. So with all the kinds of GPS and the mobile health technologies, if you're carrying your smartphone around with you, in fact, you can be followed wherever you go all the time, and there can be air measurements that are being made at the time. Now, they may tell you that there's this much PM 2.5. It doesn't necessarily tell you what the composition of that PM 2.5 is, which is a whole nother issue.
So I wore one of Dr. Anderson's bands for a week, in a small study that Environmental Defense Fund did, and found, much to my surprise, not really, that it was picking up caffeine metabolites right from my skin. Do that every day. But also things that I wouldn't have expected, like galactoside, which everybody in that little pilot study had picked up in their band. And we don't think about that very much, but there are a lot of things in cosmetic products and personal care products that, it was a little humbling for me. I hadn't thought about that very much. And some of these products will say fragrances, but they don't say what they are and what the vehicles are. So I thought it was actually not only something that could be promising in terms of doing research, but also just helping people to understand what's there, and perhaps, the kind of thing that an agency like EPA or FDA might want to know in terms of, if 100% of people are exposed to this, what do we know about it? And at least galactoside, I looked at that because I was curious about that. Not a lot. It's kind of interesting. So at this point, what I'd like to do is kind of open things up for the audience to be able to participate. Because we are webcasting, we need you to do two things. One, go to the mic, which some of you are starting to do. Two, say who you are before you make a comment or ask a question. And I would appreciate also if you want to make a comment, to make it a very brief comment, because there are a number of people who want to participate. And so I can see that our first person to the mic is Dr. Melissa Perry. She happens to be chair of the Department of Environmental and Occupational Health at the Milken Institute School of Public Health, works with Dr. Zoda. Welcome, Melissa. Hi.
Thanks so much. Fantastic panel, very, very thought-provoking, very thoughtful. I was very energized about the multiple comments related to studying preconception, and was hoping that we could unpack it a bit further in what have the obstacles been. We're talking about what happens not only during pregnancy, and you can see there's been historically a strong emphasis on what happens during pregnancy, but also at the time of fertilization. So you can have a defective sperm and actually an oocyte that is effective at repairing DNA damage, for example. So certainly we can do that in animal models, but how about in population studies? Dr. Birnbaum, you mentioned that unfortunately there wasn't a preconception cohort that was part of your children's center model yet. But the question is, what do we need to do what have the obstacles been to setting up those human studies for studying preconception, and how can we overcome that?
Well, I think Rebecca basically talked about one of them, is that only 50% of pregnancies are planned. That's number one, so people don't know. I think we've actually been looking at, in our children's and our longitudinal cohorts, the idea of looking at the next generation. Now, part of the issue is we'll know who the moms are. I'm not sure it's always going to be easy to know the dads. in the National Children's Study, which no longer exists, there had been an effort to recruit partners before pregnancy, and it was very, very unsuccessful in that. But it can be done, as Jermaine Buck-Lewis has shown. She has recruited, but a cohort of 1,000 or 2,000 people, it probably doesn't have the power to ask some of the questions we want to look at. But we do think there may be some opportunities with these million-person cohort that's going to be formed with precision medicine, under this precision medicine initiative, to do some questioning there. We're hopeful that with some of the echo studies, again, there may be some opportunities there. But it's a very difficult, it's a difficult time to recruit people for these kinds of studies. But I think it can be done. I think it can be done, but it has to be done well.
Yeah.
Hi, I'm Jerry Poggi. I teach environmental occupational health policy here at GW. I kind of would like to get reflections in a different direction. Improving children's health, we know, is going to be contingent upon the vast flow of new research, but it also is going to have to involve the families, the communities that surround them. So I'd like to get some input on how do we improve our translational research into serving communities. I know the NIEHS has been a long-term supporter of public health partnerships. Do we learn anything from those partnership activities that could accelerate the partnering between those who are doing basic research in this area to solving the problems for parents and their children?
John, do you want to start with that in terms of getting the information out there, I guess, is a piece of it, which you've certainly been involved with. Sharing the information on the web helps communities considerably, I think, the right to know issues.
Right, so go to the ACE website, at least you'll know the data that we have there. Yeah, I don't know what else to suggest, but that's a starting point.
I mean, I do think that the NIEHS centers that have very strong community engagement components, and in some cases where the members of the community are actually an active part of the research teams.
That's actually not in some. In all of our centers, where there are environmental health core centers, our breast cancer and the environment centers, our Superfund centers, I'm forgetting all of our children's health centers, any of our centers.
Everyone.
All of our centers have a community, not an outreach, but an engagement core, where from the get-go, community members are key participants and investigators in the studies, because no one knows the environmental concerns like the people in a given community.
Right.
And I think the media is another important component, right? So the media not only increases scientific literacy about these issues, but is another, important vehicle for disseminating our research. And I also think, to the extent that we can publish in open source journals, makes the information more available to a broader audience, as well as partnering with people in the NGO community, who are very skilled at leveraging the knowledge that we produce to create change.
So Lynn mentioned briefly our partnerships in environmental health, which is our overarching or our umbrella program that basically integrates all the community engagement cores and has them working together. They've got a very nice website with all kinds of tips and information on it. All of our community engagement cores work in the community and develop materials that are shared with members of the community and have a very strong educational component, including efforts related to K-12. Because I think we all know that scientific illiteracy in our country is, well, I don't want to use a not nice expression. Let's just say it's really, really terrible. And you've got to start getting to the children early. And so the K-12 is very, very important to get them involved.
I think also working with clinicians, and I think Dr. Clifton, some of the kind of work that you've been engaged with, where you're directly looking at a medical practice, well-intentioned, and founded on good science, good understanding, but it just happens that this practice doesn't work. There are many things that practitioners could be engaged with in terms of helping with prevention, helping with managing exposures, helping to educate. We do know that many people trust their doctor as an authority about science more than they trust the government, for example. And that's, survey research tells us that. But especially the obstetrical community hasn't been as engaged as it should be in beginning to understand these things. They're trying to come in, but it's, this is an area I think where we can reach a lot of communities through the medical community.
So I have to say something. ACOG, is the American College of Obstetrics and Gynecology, has been a leader in the field of dealing, and so has the American Society for Reproductive Medicine. Those are two groups that are kind of leading the field along with the American Academy of Pediatrics related to environmental exposures. Obviously, there could be more information out there. Certainly, I'd like to see the family practice docs and the internists getting involved as well, and I can tell you it's hard to make some of those inroads. Although, that doesn't mean, but clearly the pediatricians and the OBGYNs at least are some of the groups that are the most invested and the most interested in getting out information to their membership about concerns of environmental.
And they've come out with several scientific consensus statements as well.
Correct.
So they want to be engaged, and we need to develop the information and, if you may, the practices that they can use to actually on the ground do that. Yes, next question.
Yeah, my name is Yutaka Aoki. I work for NHANES, and I wanted to comment on Dean Goldman's first question about how we study BMI. How to study chemical exposure effects on the body weight and so forth. But just before I go to that point, in terms of the enhanced participation rates has been going down a little bit. So if you could help maintain the participation rate so that important data could be kept collected. But my point was about ENHANCE is that there were some additional resources that could be used for the kind of research. One is ENHANCE collected and also have some materials available for genetic data. So that's something kind of underutilized resources. And another point I wanted to make is that, ENHANCE, of course, collects body weight and other body measurements, but the kind of study we could do typically is cross-sectional. And the question and the concern is the chemical cause the weight to increase or weight to increase cause the other. So there's actually a research for this resource to do some longitudinal analysis using ENHANCE data. Using ENHANCE data as baseline, and participants could be followed up, actually passively, for mortality, their death. We have some data mechanism to use such data. Also for kids, especially, there's a linkage to Medicaid data. So Medicaid, I don't think there's a body weight or body measurements data from Medicaid, but there may be some clinical outcome that's very closely related to obesity. Some like a cholesterol measure, pre-diabetic condition. So that's a sort of one way to do longitudinal analysis linking exposure to obesity-related health outcome. So I just wanted to promote. Sorry, self-promotion.
Thank you for that. Thank you for that. I don't think it requires a response, but does anybody want to?
I just wanted to say, we've actually leveraged the fact that there's genetic data available, and my colleagues actually were able to get the DNA samples from ENHANCE participants and measure telomere length in them. And so from that, we were able to publish some of the biggest studies to date on environmental chemical exposures. And
now wait, you need to tell them what's a telomere and why is that important.
Yes, so telomeres are the ends of DNA material at the ends of chromosomes, and they help promote chromosomal stability. And as the cell divides, telomeres shorten. And once they shorten past a certain length, it leads to cellular death and impairment in function. And so telomere shortening is a risk factor for cardiovascular disease and chronic disease. So it seems to be a a biomarker of various environmental stressors, including some of the chemicals we've been talking about today.
And obesity and stress.
Right, yes.
And stress. Yes, Dr. Hu, Valerie Hu.
Right. My name's Valerie Hu. I'm in the Department of Biochemistry and Molecular Medicine here at GW. First, I want to start off by thanking Dr. Birnbaum for an excellent talk, as well as for her and the EHS's support of our research on environmental contributors to autism. My question is directly to Dr. Birnbaum with respect to what you said about the association between high molecular weight phthalates and later puberty in females. I'm wondering if there was a sexually dimorphic effect with respect to the exposures to those compounds. Is there a difference with respect to puberty in males?
Unfortunately, that study only was dealing with girls. Because it was part of the breast cancer and the environment program. So it had recruited a cohort, well, it recruited 3 cohorts, different ethnic groups in different parts of the country, of girls when they were about 6 or 7, and is following them over a 10-year period. So many of them are gone through puberty now, and they were being followed for all kinds, and lots of biospecimens taken, and so on over the time. So there were no boys in that cohort, and I'll tell you, it's actually kind of frustrating because a lot of the cohorts are only girls that we have, and a lot of the children's cohorts. And not only do dads matter, but boys matter too. And among the only studies that I can think of with puberty and environmental chemicals in boys are some of the studies that Russ Hauser and his colleagues have done in Russia, where they have shown some associations with some persistent organochlorine pesticides and time to puberty in boys. So, I think that's a real data gap.
Yeah, that's interesting because, as you know, most of the studies on autism have been done in males. So it's going to be hard, with males. So I think it's very important, especially when we're dealing with endocrine disrupting compounds in the environment as possible contributors to autism, that we need to look at the effects on both sexes.
So we have a number of cohorts of young children who are, we have, especially at the University of California, Davis, we have several studies there where they started out initially recruiting young children at the age of 3, which at least kind of traditionally has been an age that you could definitely say a child was autistic or not autistic. And they compared these, been following these children and comparing them to normally developing children and then children with other kinds of developmental delays. And then what they've done is if you have one child with autism, you have a 20% chance for each subsequent child that they might fall on the spectrum. And some people will say that's a clear evidence for the genetic contribution, which may be true. It's also, I would say, a clear evidence for environmental contribution, because in general, people don't necessarily pick up and move when they have one child with autism to something else. So there may be similarities there. And so we're following now, and there we have some information on preconceptional, both partners, for example. We have information on the second child, and then we're going to be able to follow them. And the reason so much of the work on autism is done with boys is because the ratio is 4 or 4.5 to 1, more boys than girls.
Right, but the other thing, aside from picking up and moving, so changing your physical environment, you don't change the uterine environment for the second child as well. Correct. So if the maternal environment is, what it is for the first child with autism, it's likely to be very similar for the second child and the third and so forth.
It depends, though. I mean, there are things that happen, such as persistent substances. The levels, the body burdens go down with each subsequent child. If your exposures are earlier, if you're continuously exposed, I don't want to get into an argument.
You need to see the paper that I just published, and we can talk about it.
Next speaker.
Hi, my name is Zoe Petropoulos, and I'm going to switch gears and ask a policy question. So since we have a new president and administration coming in less than a year, hypothetically, if you got to sit down with that president, what initiatives or what efforts would you want them to put in during their tenure to advance this field and also to improve the translation of the findings into interventions and public health programs and policies? What would those really, think high level, so would you want something like Cancer Moonshot or the Precision Medicine Initiative, or just more funding? What what would be the most high impact policy change? And then is there something out of Congress that you would want changed as well, more than just maybe passing a new TSC reform bill? What would you need or what would you want?
So I don't know if the members of this panel are all, prepared to address that, but I'm going to go ahead and lob it to them to see what they'll say. Starting here.
I think I better pass on that one.
You'll pass.
Well, I mean, I think in addition to how EPA regulates toxic substances, a thorough review and changes in food, we were talking about this before, what FDA regulates and how they regulate both the food, food is a huge, very important to people. People are very concerned about what they're eating, but I think there's a lot of potential for exposure there that a lot of people don't have control over. As well as cosmetics, another big area that impacts everybody that's largely unregulated right now. I would also want attention to our nation's most vulnerable communities, because right now they really do often carry disproportionate burdens, and these disproportionate burdens are not adequately taken into account when new decision making is made about where industries are placed, or what infrastructure is updated, and those other types of decisions. So those are some areas that I think it would be good to have higher level interventions.
Rebecca, did you want to?
I would just add to, in general, increase funding. I think that is probably the key. And then, to target specific populations, what Dr. Zota was talking about.
So I would say money, because we know that with more research we can make better decisions. But I do want to stress that regulatory change and policy changes have a much greater impact at a much lower cost than, for example, personal decisions or medical decisions. There was a study that David Bellinger and co-workers did a couple of years ago where they compared the cost of medical treatments to policy change. And again, the policy changes are much more effective. It's very hard to make people change their behaviors. And yet policy change, it's hard to get policy change too, but that is where I think effort should be put to make some changes. And Ami just alluded to the fact of unfortunate substitutions, which is the polite way of talking about whack-a-mole or the chemical conveyor belt, which is what tends to happen is one chemical is shown to be problematic, like some of the high molecular weight phthalates. So then they come up with ones that are a little lower molecular weight, and guess what? They're not tested, so okay, they're fine, but when some people start studying them, they turn out to be just as bad. And this is happening with a whole series of different kinds of substitutions. So I'm sure everyone in this room got rid of their Nalgene bottles that had BPA in them because of concerns with BPA. But now we're finding that what's in those bottles now may be every bit as bad, if not worse, than what was put in than the BPA was. So you jump from what you know is bad to something that you know less about. And I think it'd be really nice that if we would test things before we expose people to them.
So I'll give a response as well. And I think that one thing that I found kind of disappointing, actually, to somewhat in terms of the cancer moonshot, has been that so much of the emphasis has been on speeding the development of new medical treatments for cancer, when yet so much of cancer is preventable using knowledge that we already have. And I don't think that we can treat our way out of the cancer problem. I think that the CDC, that for a long time has said 30% of cancers could be prevented if we applied what we already know. And Dr. Birnbaum showed you the enormous annual costs of environment-related disease across the world and in the United States as well. And so my mantra would be prevention, prevention, prevention. But it's not that simple. We need research to inform prevention. We've talked a lot in this country about translational research, and the precision medicine initiatives about translation, basically, of what we know to medical treatment and personalizing medical treatment. But I think that we also need research that helps us to translate to better population and public health
So Lynn, I'm going to jump in because it's really important to understand that the National Cancer Institute now has put out a call for people to comment on the Cancer Moonshot, and they're looking for suggestions of what should be done. And I've been talking to Doug Lowy, who's the acting director of NCI, and talking about the need to have prevention as one of the major goals of the Moonshot. So I think if people, put that into this call for information that NCI is looking at, it'll be a way to at least hopefully get them to address that issue.
And somehow the deans of the schools of public health all wrote a letter too, and any of you also can contribute and can help to shape this. But this is a policy of this administration. So I guess another message that I would have has to do with the need to have strong continuity of some of these initiatives from one administration to the next. And the fact that one of the things that has certainly plagued efforts like the National Children's Study has been, with changes in administration, you have support and then you don't have support. You can have management of a big study like that flipping over. And I think that we see, and especially with Linda's engagement, the ECHO study is starting to take shape. There's a new director. But will a next administration stay the course? Will they own it? Will they care about it? Will they move it forward? This is something that all of us need to be aware of, so that if they ask us, what do you think, that we would certainly say, yes, stay the course with some of these things, because it's always tempting for new people to want to create their own thing. That it's called something different, looks like something different, that is theirs. We have a next question.
Hi, Jennifer McPartland. I'm a senior scientist in the health program at Environmental Defense Fund. So throughout your presentations, many of you gave really great examples of specific environmental exposures that we have come to understand are associated with various health outcomes. But I'm sure also all of you know that the vast majority of chemicals out there in the marketplace are very poor, are very data poor, and you've already started to exemplify that with the situation of regrettable substitutions. And so I'd like to ask all of you, how do you think, and maybe this might be directed to Dr. Birnbaum first, these new novel emerging methods like the exposomic centers coming out of CHEER, as well as the predictive mechanistic-based approaches coming out of Tox21 and NIEHS could help move the ball forward in terms of filling those data gaps, while also simultaneously appreciating the biological complexities of the very health outcomes that we're trying to predict chemicals maybe have an effect on.
Okay, so that's kind of a multi-part
Yes.
question.
Guilty.
So you talked about the exposome and you talked about the Tox21 and how these could be integrated or used together. So Tox21, for those of you who are unfamiliar with it, is a cross-agency effort that involves two parts of NIH. It involves NCATS, which is the National Center for Advancing Translational Science, and NIEHS. It involves EPA's Office of Research and Development, and it involves FDA. And what this is doing is developing rapid, high-throughput screening and also mid-throughput, different approaches, so that we can test more chemicals more rapidly. The maximum number of chemicals that are, say, tested in experimental animal models in long-term cancer studies is about 8 to 10, which is a very small number of the 80,000 or so chemicals out there in commerce, about 60,000 of which we know nothing or virtually nothing about. With some of the approaches, I used to not be a believer in these high-throughput in vitro kind of approaches, but when instead of running 10 chemicals or 100 chemicals, you run thousands of chemicals, and instead of looking at 1 or 2 or 3 different kinds of endpoints, you can look at 600 different kinds of assays or 700 different kinds of assays. You begin to see patterns evolve. And in fact, what you can see is that looking, and we've looked at over 10,000 different individual chemicals, and what you can see is that maybe 10 to 15% of them are what I would call the bad actors. 20% of them, 15 to 20%, do nada in anything. And the rest do a little. Well, where am I going to put my effort? I'm going to focus on those ones that appear to be the bad actors in some of these high-throughput studies. And I'm going to move to doing some of the mid-throughput approaches, like using worms, C. elegans are using things like Drosophila, or for vertebrates using zebrafish, which are one of my favorite models, where in fact you can run hundreds of individual fish through hundreds of different chemicals and begin to understand what's happening in their endocrine system. Are you messing up development? For those of you who don't know, zebrafish, for their first 5 days of development, and their whole life cycle is only 3 months, they are totally transparent. So you can watch the heart forming, you can watch the brain forming, you can watch, it's absolutely an amazing thing to look at. Now you have to do it with a microscope because they're really little, but that's okay. But I think that those are some of the kinds of approaches that we have to use. We also are doing a lot of computational modeling to identify factors which may be problematic. And as we were talking, or as I was saying with the unfortunate substitutions, when you move from one chemical to another, because you don't want to have to change the industrial process, so you just change one functional group or something, why would you think, if it doesn't change the industrial process, why would you think that's going to not change the biology? There's kind of, we don't always think through what we're doing. I would also suggest that a chemical that is biologically, especially highly biological active in many different kind of assays, is likely to cause lots of different adversity. It's going to be a function of when did the exposure occur, like during what susceptible window, was it acute or was it chronic, and what were the doses that you used, plus what's the entire matrix that you're dealing with. What's happening, what's going on in the diet. We know, for example, that if you expose experimental animals to chemicals on a low-fat diet, you get a completely different response than if they're on a high-fat diet. Well, what does that say about the American public, where most of the American public eats a high-fat diet? You can begin to look at, but I think these are the things that we're beginning to look at. And I think we're going to need some very complicated computational approaches to understand it all.
Mm-hmm. Thank you. Thanks. One last question for this session. Dr. Pelfrey. Hi, Kevin.
Dr. Pelfrey, George Washington University. And one comment and then a second comment / question. I have been involved in autism research now for 14 years. And it occurs to me, no one's ever asked, what would you ask for from the president if you could? And so how happy I am to be here where that's a posable question and the president might listen.
Could we ask you that question?
No, but so it's really extraordinary. So I'm glad to be here. And then we've been doing research. We know autism has been a very genetics-heavy field, and part of that is the history of the vaccine controversy and blaming parents for environmental insults caused by bad parenting, and then the vaccine vector sort of coming in and really killing all innovation in environmental work around autism because it's so hard to get it funded. And then ramping up, the genetics, and also with the sort of genetic revolution, that work has been heavily emphasized. And it occurred to me as people were talking, we now know our major contribution to the science of autism is that about 20% of autism can be accounted for by de novo mutations that are found in children, usually copy number variations or single nucleotide variations. And so they're de novo, and that's exciting to we who are in genetics because we can start to trace the pathophysiology. And it's been adequate to just call them de novo. And then it occurred to me, no one has ever asked in as many meetings as I've ever been to, well, what caused the mutation? Right. It's just enough that it's de novo. And so there are actually thousands of subjects at this point where we've characterized carefully everything you can about the parents, their current psychological state, about their genetics, about their brain. And we've never asked a question about anything related to environmental exposures. And many of our grants are funded towards identifying the next member of the family born to see, are they going to develop autism, infant SIB studies, this is called. And I'm stunned by the fact that we haven't taken the opportunity to do essentially birth cohort designs where we know the genetic lesion that's de novo, and we can say, is there going to be another one in this child? And then what is the cause? And we can follow the parents preconception. And the parents will be very willing to participate. They'll wear anything you want and do anything you want because of the return is that their child is so closely surveilled. So more of a comment, but I think what I've noticed in the policy world of this, sort of sitting on the Interagency Autism Coordinating Committee, the voices of those studying environmental impacts are quelched. You know, what we do here is from parents often talking about vaccines, and we scientists sort of zone out at that point. But there's something beyond that I think I'm really impressed by the amazing science presented here today, and I think something that GW could be uniquely strong in, because we already have access to this data, and now we have access to this amazing Institute for Public Health. So, long comment. Sorry.
Well, we'd love to open a dialogue with you. I sit on the ICC also. I don't always get there, but to those meetings. But we've ramped up our autism portfolio dramatically. I'd say over the past 6 or 7 years, going from only about $1.5 million a year to now about $12 million a year. We've had a couple of FOAs out there. Those are funding announcements related, and we do are funding a couple of SIBS kind of studies where some genetics are being looked at in those studies, but much more focused on environmental. And I think it'd be great if we can get together and cross them, because that's what we need.
So that's really the science is going to be at the interaction. We still haven't found a gene for any neuropsychiatric disorder specifically. And so it's always about the interaction.
That's right. Thank you so much for that. And thank you so much to our panel, and especially to Dr. Birnbaum for what's been a wonderful session, and to those of you in the audience who participated in the discussion. It's been great. I think we're going to have
No, if you could give a great hand to the team here. I mean, Lynn and ICF and others who helped put together a great panel today. If you could give them all a big hand. But on that note, after listening to Lynn and Linda, I'm questioning if two of them were separated at birth somewhere. But thank you all again. Appreciate you coming today.
Thank you.